Hi, everybody. Thank you so much for coming today on a Friday afternoon. It's a really good turnout. I was worried that everybody would be gone already. So before we start today, I just wondered if there was any new people that are here that we can introduce.
Should we come up there?
No, just stand up and say your name and like what lab you're in and where you're from.
Hi, I'm Finn, in the Predator Prey Lab in Agro-ecology.
Hi, Finn. Thank you.
Oh, we're
That agro-ecology lab, an intern.
Cool.
In the fish lab, I'm from upstate New York. My name is Caitlin, I'm also in the fish lab, I'm from New Orleans.
Great to see all these new, fresh faces. All right, my name's Betsy Boughton. I'm the program director of agroecology at the ranch. Today, we're going to have an intern presentation by Christiana. Claire was co-advised by Dr. John Xiao Chu at University of Florida, and she also had a lot of support from our research assistants and other people in our lab. So Haley Henderson is a research assistant at UF that was very supportive, Emily Anderson at the ranch, and also Lydia Landau, who no longer works with us. But it takes a village to support a research intern, and I think Claire had a good village around her. Claire arrived at Buck Island Ranch in May 2022, so she's been here about 8 months now. And before that, she graduated from Siena College in New York in May 2022 with a BA in Environmental Studies. And she was a policy intern at the Citizens Campaign for the Environment in Connecticut. And that's where she worked on pollution, clean water, and drinking water funding opportunities. But she wanted to come here and get a taste of research. After her internship here, she has accepted a position as an environmental lab technician in Connecticut, and she's going to be analyzing air, soil, and water samples. And I think a lot of what she did in her internship set her up for that position. So we really enjoyed having Claire in our lab, and we're going to miss her. And she's very hardworking and steady. And thank you so much, Claire, for all of your work at the lab. Now I'll turn it over to you. And Claire's title is The Impact of Precipitation Manipulation and Grazing Intensity on Soil Greenhouse Gas Emissions in a Subtropical Grassland.
Betsy. Hi, everyone. Thank you all for coming to my talk. I'll be presenting my final research presentation. So Central Florida provides an exemplary location for climate change research. Subtropical grasslands are important for greenhouse gas research because they provide many ecosystem services and are likely to experience future climate variability driven by changes in precipitation and extreme flooding, both of which are catalysts for driving large emissions from underlying soils. There are extensive research and climate models for the implications of greenhouse and gas-induced climate change on Florida. Not only is the magnitude of precipitation changing, but the timing and seasonality of precipitation events are shifting, which are causing harmful effects to agricultural productivity, ecosystem services, and even species that inhabit narrow ranges. On the right is a study that was done in 2011, showing that there will be a seasonality shift, which means that warmer months are getting warmer, and there's a large variability in precipitation. I chose to study carbon dioxide, nitrous oxide, and methane because they are the three main greenhouse gases that come from subtropical grasslands. Carbon dioxide and methane are also rarely studied simultaneously. Carbon dioxide is the main driver of climate change caused by direct human influence. It has a global warming potential of 1, which means it stays in the atmosphere for a very long time. Global warming potential is a measure of how much energy the emissions of 1 ton of gas will absorb over a given period of time, relative to the emissions of 1 ton of carbon dioxide. Nitrous oxide emissions result from fuel combustion when nitrogen in the air gets oxidized. Nitrous oxide has a global warming potential around 273 times that of carbon dioxide for a 100-year time scale. Lastly, methane has a lower global warming potential of 25, but also absorbs much more energy than carbon dioxide, and methane has a shorter lifetime of 11 years. These three gases are largely driven by soil moisture, which is an important variable for seasonally flooded ecosystems like subtropical grasslands, which are understudied compared to temperate regions. They also differ from temperate ecosystems because they contain well-drained mineral soils, which create oxic conditions below the water table, influencing mechanisms of greenhouse gas flux. So it's known that flooding and precipitation influence greenhouse gas flux because the effect of moisture on oxidation and bacterial composition of soils. For example, researchers in Brazil found a close correlation between soil moisture and methane fluxes, describing that the highest level of methane production and oxidation occurred during the rainy season. Soil moisture also has a significant diurnal variability, so it can be difficult to quantify, which could explain some inconsistencies between studies. A similar study, which took place at Archbold's Buck Island Ranch, was conducted over a 3-year period, and soil and flux samples were taken in a fenced improved pasture. The authors concluded that flooding caused sustained effects on methane fluxes. And they also interestingly observed a delay or a lag in methane fluxes, and emissions peaked as the water table receded. Cattle grazing is also a crucial factor, and it's important for improving the structure and function of grasslands, but it also has some interesting effects on flux. For example, Gomez Casanovas found that grazing increased total net ecosystem methane emissions and removed above ground biomass, but it increased the net storage of carbon, increasing the net CO2 sink strength. Lastly, pasture management dramatically influences grasslands. In De Lucia's experiment, methane emissions were found to be higher in improved pastures due to increased soil moisture, temperature, more extensive ditching compared to semi-native pastures. So these were my 4 research questions. I first asked, how does pasture management intensity affect soil emissions of carbon dioxide, nitrous oxide, and methane? How does grazing intensity affect soil emissions of the 3 gases? How do changing rainfall patterns affect soil emissions of the 3 gases? And lastly, do these 3 drivers interact with each other to impact soil greenhouse gas flux? So all of my sampling took place at Archbold Biological Station's Buck Island Ranch, which is a 10,300 acre cattle ranch that operates as an ecological field station. I sampled 3 improved pastures and 3 semi-native pastures shown in this image. So I was interested in studying greenhouse gas fluxes in 2 different pasture types because of the unique characteristics that distinguish both of them. For example, improved pastures are beneficial for maximizing beef cattle production. So they have a history of fertilization, more extensive ditching, and they are grasslands that have been converted to non-native C4 forage grasses like Bahia grass. Semi-native pastures, however, have a lower cattle load, they've never been fertilized, and they have a mixture of native grasses like Andropogon and Panicum species. It's important to mention that the improved pastures are occasionally fertilized with nitrogen, and they were annually fertilized with phosphorus 15 to 20 years before 1987. So although fertilization on the ranch has been reduced, there are prolonged effects of these nutrients on plant biomass, species composition, and even surrounding pastures and wetlands as proved by De Lucia 2019. So this is the rainout project at Buck Island Ranch where I took my samples. The project began in 2021 and it's expected to continue until 2026. It was funded by NIFA to Dr. Zhengjiao Q at UF in collaboration with Dr. Betsy Boughton. There are 2 pasture type treatments, 2 grazing treatments, and 5 precipitation treatments. This is a diagram of the project experimental design. The diagram on the right shows the treatments that are in each of the 8 pastures. And these treatments are based on the historical range of variability and the determined levels of 50% change in the wet season and 75% change in the dry season. In increased quantity, the wet and dry season get wetter. In reduced quantity, they get drier. In enhanced seasonality, the wet season gets wetter and the dry season gets drier. Decreased seasonality or diminished seasonality is the inverse, and control gets left the same. So for sampling event 1, I sampled both high and low grazing treatments, but due to the amount of labor, I decided to scale my sampling down to make it more feasible. So I sampled from only the high grazing treatments for sampling event 2 through 5. Above are the stocking densities for each of the treatments, with improved having a higher density to account for increased plant productivity. This is a more detailed diagram of what the treatments look like, created by past RA Lydia Landau. The rainwater gets diverted through a gutter, and then it gets pumped into the sprinklers of the adjacent treatment. So these were my hypotheses for the effect of pasture type on greenhouse gas flux. The green arrows represent positive fluxes and the red represent negative. The number of arrows represent the magnitude or the strength of the fluxes. The more arrows represent a large expected increase due to the design of the static chamber and results from similar studies. So the control is one arrow rather than steady. I first hypothesized that fluxes would be greater in improved pastures than semi-native because fertilizers stimulate both biomass productivity and microbial activity, which increases CO2 and CH4. Improved pastures also have a higher stocking density, and I hypothesized that there might be lower or even negative fluxes of nitrous oxide in improved pastures because in some cases, denitrifiers in the soil use nitrous oxide as an electron acceptor when oxygen is limited. And I have one arrow for each semi-native pasture because of lower soil moisture levels, no history of fertilization, and they have a lower stocking density of cattle. For grazing, I expected low grazing treatments to have higher carbon dioxide fluxes because there would be more above ground plant biomass, while in the higher grazing treatments, there would be less photosynthetic surface area. Grazing also removes dead buildup and stimulates root and microbial activity. Studies on the effects of grazing on nitrous oxide have been somewhat inconclusive, but I expected negative fluxes in the low grazing treatments because these treatments have been proven to reduce more litter. I just lost the cursor. That one? The cursor.
It's on the projector screen, so just move it slowly to the left and right and show
There it is. Oh, perfect. Okay. Did I leave off of grazing? Precipitation? Go. Yeah. Keep going past grazing. Yeah. Okay. My hypotheses for the effect of precipitation treatments were mostly supported by previous studies on soil moisture and greenhouse gas flux. I expected lower or even negative fluxes in nitrous oxide due to the reduced quantity and diminished seasonality plots because of uptake of nitrous oxide in soils have been reported for grasslands in several studies. And I expected this especially in semi-native pastures where there might be a lack of mineral nitrogen. So for my methods, I use the static chamber method for my project because it's ideal for sampling various treatments across a small area. The static chamber method is widely used and involves sampling from multiple closed chambers at specific time points to measure gas concentration over time. And the Collier et al. Protocol was the main source that I used, but Sullivan and Hart 2015 was referenced for guidance on how to best install the collars and store the samples. For my research design, I installed the collars 3 centimeters into the ground in each treatment and clipped the vegetation to the height of the chamber. Then I took ancillary measurements and began sampling 1 hour after I installed the collars. At T0, I started a stopwatch, attached and sealed the lid on the chamber, and collected the ambient air sample immediately. The rest of the samples were taken inside of the chamber, with T1 at 15 minutes, T2 at 30 minutes, and T3 at 45 minutes. So I sampled each of these 5 treatments at each time series to end up with 4 samples for each treatment for all 6 pastures in each sampling event. The samples were then sent to the UF-IFAS Fort Lauderdale Research and Education Center to be analyzed using gas chromatography and to determine the concentration of the 3 gases. So I had 5 sampling events. Sampling event 1 through 3 took place before grazing, and 4 and 5 took place after the grazing treatments in October. And I waited a full week after the pastures were grazed to sample again. The timeline for my sampling was bi-weekly before and after grazing. And the bi-weekly schedule is based on when each individual pasture had been grazed. There was a delay in grazing in the experiment due to the hurricane in September, but luckily this occurred between sampling events. We removed the shelters during the storms. The shelter design does not fully account for extreme rainfall or non-vertical rainfall events. So here's how I calculated the flux for each of my chambers. First, I had to calculate the slope by running a regression for each of the time by gas ppm data, and I'll show an example of this on the next slide. I then had to do 2 more conversion steps to convert the slope of the flux with the units of microgram per square meter, meter per hour, and the equations are not defined here, but I'm not going through all of the details today. So this is an example of some of the fluxes. On the left is an example of a positive carbon dioxide flux from improved pasture J, high grazing, increased quantity. And on the right is a negative nitrous oxide flux from semi-native pasture H, low grazing, reduced quantity. So these were my statistical analyses. For carbon dioxide and methane, they were log transformed to achieve normality. Nitrous oxide fluxes were very small and were non-normal. To assess pasture type and precipitation treatments, we used the analysis of variance. To assess grazing impact, we used analysis of variance on just the August sampling, and the Kruskal-Wallis test was used to assess nitrous oxide. So now onto my results. On the x-axis is the soil moisture, and on the y-axis are the fluxes. The graph on the left shows methane fluxes were positively correlated to soil moisture, and the 2 graphs on the right show that nitrous oxide and carbon dioxide had a slight positive correlation, but were not significant. And we were surprised that we didn't find a significant relationship of nitrous oxide fluxes and soil moisture, but this could be due to the high variability of soil moisture and temperature throughout the day. On the x-axis is the sampling event, and on the y-axis is the CO2 flux. The blue bars are improved pastures, the black represent semi-native, and we found that soil CO2 emissions were greater in improved pastures, which is supported by previous studies. Next on the x-axis is sampling event, and on the y is methane fluxes. The blue bars represent improved pastures and the black represents semi-native, and there was no clear effect of pasture type on methane or nitrous oxide fluxes. On the X are the grazing treatments, on the y-axis are the CO2 fluxes, the blue are improved and the black are semi-native. And this graph is showing data from sampling event 1, where I took samples from both grazing treatments. And you can see that carbon dioxide emissions were greater in high grazing improved pastures compared to low grazing improved. That was not the case for semi-native. So if you look at both black bars, fluxes were slightly higher in low grazing semi-native than high grazing semi-native, which past research at the ranch supports. And here the x-axis is the precipitation treatments, and the y-axis shows soil moisture. The colors of the precipitation treatments are explained in the key on the right of the slide, and the left section of this graph shows results of the improved, and the right shows semi-native. And you can see that there was no clear effect of the precipitation treatments on soil moisture. Although it tends to be lower in reduced quantity, it was nothing significant. And there was also no effect of the precipitation treatments on the soil emissions of greenhouse gases. And this could be due to the fact that the rainout experiment has only been running for about 1 year. So to summarize my results, fluxes varied by sampling event, and sampling events had variable weather conditions. Methane fluxes were positively related to soil moisture. Carbon dioxide fluxes were greater than methane and nitrous oxide, and CO2 is easier to analyze and more responsive to treatments. Improved pastures had greater carbon dioxide flux than semi-native. Grazing intensity increased carbon dioxide fluxes in improved pastures. Precipitation treatment impacts were not detectable on fluxes or soil moisture. And nitrous oxide and methane didn't respond significantly to any treatments. So my discussion, understanding the mechanisms of greenhouse gas flux is understudied and could be important to microbial activity. Improved and semi-native pastures also have different species composition and different soil characteristics. Future studies could explore the patterns of which variables associated with grazing influence fluxes. Lastly, for precipitation, experiments can be difficult to control flooding. The rainout experiment hasn't been running as long, and the samples were only taken over 4 months in the wet season. So future studies could run longer, and we might be able to see more of an impact during the dry season. So the significance, new data from this project will improve predictions on future dynamics of low latitude grasslands, and new science-based knowledge will inform sustainable land management to foster climate regulation service of grasslands used for beef cattle production. My research advances mechanistic understanding of drivers, processes, and complex biotic and abiotic interactions underpinning greenhouse gas flux in managed grasslands. And lastly, my research allows us to develop and assess land management strategies and adoptions that can mitigate effects of climate change and improve climate regulation service in grasslands. And it's been an amazing 8 months, and I'd like to acknowledge Dr. Betsy Boughton, Dr. Zheng Hu, Haley Henderson, and Caroline Navarro, everyone at Agro-ecology, Shafali, Greg, Amartya, how you, Mandy, Carly, and Alma, Emily and Lydia, all of my housemates, Akeem, Luca, Kyle, Alana, Fern, and Jake, Archibald, the University of Florida, Eltar, and the grant that funded my research, NIFA. These are my references, and we can take some time for questions. Yeah,
That was a really great talk.
thank you.
So I'm not a biologist, but can you explain why nitrous oxide and methane did not show any responses? Like, is there something chemically different about that?
Yeah, it's tricky with nitrous oxide especially. There's been studies that have shown positive and negative fluxes in both wet and dry soils. So I don't know much about microbial activity, but I think it has mostly to do with those mechanisms. Jeff?
So you had as one of your results that grazing intensity changes the carbon dioxide fluxes. But that was for the first sampling event, and that was before these pastures were grazed, correct?
Yeah, they have been grazed previously. How long ago? Was it a year ago? What was the last treatment?
Yeah, it was a whole year that they were rested from grazing.
Yeah.
So, I mean, do you have any ideas for how that would or why that would impact?
Yeah. I think
Don't
that's
forget to repeat the
Oh,
question.
sure. So the question was, how does the previous grazing treatment from a year ago affect grazing treatments from before? So I guess that's a good point, that it's not a clear, steady control that we're starting with. But I guess that it also makes sense because the surrounding pasture is also being grazed and has cattle in it. So we wanted to make sure that the differences between improved and semi-native were accurate. The higher stocking density and soil moisture were kept the same. That's a good point.
I was curious, so if you do this study like in more northern latitudes or southern latitude, would you expect differences in the emissions, like based on where you did the study?
Yeah, I think
Do you want
so.
me to repeat the question?
Oh, sure, yeah.
So if you did this study in more northern latitudes or southern latitudes, would you expect different fluxes?
Yeah, I did measure soil and air temperature. But I don't think we analyzed them as thoroughly as the fluxes. But I think higher temperatures definitely have higher fluxes. I think especially for methane. Right.
Yeah.
Do we have a clear explanation for why the CO2 fluxes were doing such different things in the semi-native and the improved pastures?
Yeah, I think past studies have shown that improved pastures have higher quality of carbon, and so it makes it easier for the microbes to digest. And then grazing also helps with that as well.
Now, so you mentioned the hurricanes. I'm just curious, like, what effect do you think that actually had on your results, or do you think that was just like, you know, kind of noise in the background and didn't really affect things overall?
Yeah, it definitely,
The question was, really bad at this. So the question was, how do you think the hurricanes affected the results of your study?
yeah, well, it definitely affected my sampling. I had to wait an extra week. Instead of just 1 week, I waited 2 weeks to sample so that the flooding could recede. And so I think that definitely had an effect on methane fluxes. Yeah, increased soil moisture increases fluxes, but I had to wait that period. So yeah, if I were to do this again, I would be interested to see what the results would look like.
And I'll also add that this type of study, like trying to manipulate rainfall and make some places drier and some places wetter, is really challenging in our area because we do get so much rainfall and it's flat, and so everything just floods when we have a hurricane. So that erases all of our efforts to manipulate precipitation. So I think it's going to be challenging to see effects in our grassland, and it might take a really long time to see effects of those precipitation treatments.
Thank you.
Yeah, Karen.
So a lot of sampling looked like it was in the fall and into kind of late fall, almost winter. Do you think you'd get the same results if you had gone through a whole year's work and had more of the dry season involved?
I think the dry season would be,
Do you want me to repeat the
yes,
question?
please.
So a lot of Claire's samplings were in the fall, and do you think your results would be different if you did a whole year of sampling?
Yeah, less soil moisture, lower temperatures. I think, I don't know if it would be opposite, but just different interactions happening in the soil.
Yeah, I also think the N2O and the nitrous oxide and the methane are really like pulses and can be very episodic. So if we did it for a longer period of time, we might be able to better catch those pulses.
And we also had a pretty dry, wet season. So that's interesting too.
Any questions online, Laura?
No.
Okay, great. Thank you so much for your